All right, welcome back, biology. Today we're going to be talking about cellular respiration. So you'll notice with the cellular respiration equation, now this occurs in the mitochondria, and we talked about photosynthesis that occurs in the chloroplast. For cellular respiration, you'll notice with the equation that the products of cellular respiration are the reactants in photosynthesis. So what cellular respiration does is it takes sugar and oxygen, and it breaks down that sugar through a three-phase process that we'll look at each phase in order to create a bunch of ATP. So that's the goal of cellular respiration. So it's taking these big sugar molecules and breaking them down and converting them into smaller, more usable energy molecules called ATP, or adenosine triphosphate. Photosynthesis was taking carbon dioxide out of the air using water and sunlight in order to build up these big sugar molecules. So it's this cycle of codependence. The mitochondria needs the chloroplast, and the chloroplast needs the mitochondria. We say cellular respiration is exergonic. So if you remember from when we talked about energy, that's going to mean it's going to release heat. Biggest thing to keep in mind here is that we're taking glucose using oxygen. Without oxygen, the amount of ATP that will be produced is very, very small. So we need oxygen to make lots of it. And we're going to take glucose and break it down, and we're going to make tons of ATP. Another really interesting thing about cellular respiration is it occurs in every living thing on the planet that's ever been discovered. This is the way that life forms on Earth are able to produce energy, ATP, to stay alive and get every cellular process just about done using that energy molecule. So when we compare cellular respiration to photosynthesis, we see an interesting codependence. Photosynthesis makes or releases oxygen, and it makes and produces glucose. And those happen to be the reactants or ingredients the mitochondria needs to be able to go through cellular respiration, where water and CO2 are released. If we look at the structure of a mitochondria, the structure of the mitochondria is very important in terms of cellular respiration. And this is where cellular respiration occurs. You'll see this interesting structure where it has an outer membrane, and then it also has this inner membrane with all these folds called cristae. When you increase the folds on something, think about an air filter. What are they trying to do, engineers, when they fold something over and over and over? They're trying to increase the surface area. The mitochondria has these inner folds, these cristae, in order to increase the surface area. And you'll see why with this electron transport chain that it is on the inner membrane. So the more folding that there is, the more electron transport chain surface area you can have to drive cellular respiration. And that specific process that we'll look at. And then the matrix is also important, and that's this space in the very center of the cristae. In between the inner space of the inner membrane is the area that we're talking about. There is a space between the inner membrane and the outer membrane, and they call that the inner membrane space. So cellular respiration, it can occur aerobically, meaning requiring oxygen, but it also has a pathway when there is no oxygen, and we call that anaerobic, meaning without oxygen. Ideally, we're going to have aerobic pathway because it makes so much more ATP than the anaerobic pathway. The anaerobic pathway is necessary because under conditions where there is no oxygen, you need some kind of process to make ATP, because without ATP, you're dead. So we do need some kind of pathway for cells in our body that are deprived of oxygen, who need some emergency ATP to get by. But it's not optimal, it's less efficient, it doesn't make much ATP. The phases for aerobic respiration we'll look at will be glycolysis, the Krebs cycle, and the electron transport chain. And if we're talking about an anaerobic pathway, Glycolysis will still occur because that happens in the cytoplasm. You don't need the mitochondria for that. And then the second process that will happen is some type of fermentation. Could be alcohol fermentation or lactic acid fermentation, and we'll look at examples of that. So here are the three big stages or processes that occur in aerobic, meaning requiring oxygen in the cell. So glycolysis again, in aerobic or anaerobic, is going to happen out here in the cytoplasm. And we'll go into detail on all of these, but just an overview: glycolysis, glycolysis, glycosugar, lysis to split. We take glucose and we split it essentially. Krebs cycle, one of the major things that needs to happen in this process is producing NADH and FADH2. Makes a little bit of ATP, and so does glycolysis, but not very much. But the real benefit to the Krebs cycle is producing NADH and FADH2 that are used in the ETC, or electron transport chain, to make a massive amount of ATP. And again, the whole point of respiration is to make ATP. So first we'll talk about glycolysis. Again, glycosugar, lysis to split. It takes one glucose molecule, breaks it down into two molecules, and the molecules it splits it into are called pyruvates. And that's going to cost 2 ATP. So you got to spend money to make money. So here you see coming into this glycolysis pathway, 2 ATP and a glucose. And we put all that in. We have to pay $2 or 2 ATP, but we'll look at the steps that occur. You get out 4 ATP and 2 pyruvates. So even though we spent 2 ATP, we produce 4. So that means we're going to net 2 ATP when we subtract out our cost. It's also going to make 2 NADHs and 2 pyruvates. And all these molecules are going to be used later on. So that's the first step, glycolysis. So you need to know where it occurs, cytoplasm, what comes in, what goes out. And they call that part where you spend 2 ATP to initially break that glucose, they call that the energy investment, energy investment phase. And then the second part, once it splits, well, you have two pathways now where that G3P is going to be modified into pyruvate. And they call that part the energy payoff, because now, right, before we paid in 2, and now we're going to get out 2 on each side for net 2. So again, just being familiar with the reactants and products in each step. So we carried the Y over here. So then between glycolysis and the Krebs cycle, there's something called the intermediate step. Sometimes they call it the link step. And this is how we convert pyruvate into a more usable form for the Krebs cycle. And it's going to be converted into acetyl coenzyme A, or acetyl-CoA for short. Also during this process, we're going to make an NADH and some carbon dioxide. So right, we always say we breathe out CO2, and plants are breathing out O2. And that's true. We are breathing out CO2, and this is one reason why it's a waste product of cellular respiration. Plants are breathing out oxygen, so to speak, because that's a product of photosynthesis. Plants also do cellular respiration. So plants are also going to be releasing CO2. The difference is the plants can use that CO2 to build sugars, and we cannot because our animal cells do not have chloroplast. So anyway, going back to this, we got glycolysis, the link step or the intermediate step, and then we get into the Krebs cycle. So here's what would occur, our products for the intermediate step. Now we've got 2 NADH because it happened twice, 2 acetyl coenzymes, coenzyme A, and 2 CO2 molecules because again it happened twice from the 2 pyruvates and the oxygen that we started with. And just something interesting about this step is that if you deprive a cell of sugar, or if there's not enough sugar, then your body can take fat or protein and actually shove it into this pathway at this point, and then use that to go through cellular respiration. So people who, they go on those no-carb, low-carb diets, Well, their body's got to get that energy from somewhere. So it's going to go to the next easiest molecules to break down, which is going to be fat and proteins. They're not as easy to break down as glucose, so they're not going to be used when sugar is available, because sugar is just so much more efficient. That's why we say sugar is that quick energy. But this could be a way to burn that fat. So then we get to the Krebs cycle. Now we're in the mitochondria. Krebs cycle is going to happen in the mitochondria. And remember, at the intermediate step, we produced 2 acetyl coenzyme A molecules. So that means they can go through this twice because we have 2 of them. Now here we'll just look at one coming through. The acetyl coenzyme A, we say 6C, that means 6 carbon molecule. As it moves through, it's becoming oxidized. And if you remember oil rig, oxidation is loss of electrons. So as it loses electrons, those electrons can be used, right? If we have NADH positive, if we add electrons to that, well, then it could become negative and be attracted to an H plus or a proton, in which case they could be put together. So the oxidation of acetyl coenzyme A is used, the electrons off of that are used to produce NADH. And you'll see as the molecule moves on, makes another NADH, keeps going, makes some more CO2, makes some ATP. You'll notice it's being oxidized, it's using electrons, it's losing carbons. And you'll see CO2 is coming off, that's where that carbon's going. And then as it continues, we continue oxidation, we get FADH2, and we get more NADH. So you'll notice that as it moves through that process, it's going to make 3 NADH, 1 ATP, and 1 FADH2. It's going to happen twice, so we can double all of those molecules. So what we end up with, again, we started with this, 2 of each, coming out of the intermediate step, and glycolysis. And we're going to end the Krebs cycle with 6 NADH, 2 ATP, 2 FADH2, and 4 CO2. This NADH, the ATP is good, yeah, we want that. But the NADH and the FADH2 is key because these are high potential energy molecules. So we're going to use them to make way more ATP than we've made so far. Right, in glycolysis, we netted 2 because we spent 2, made 4, so we netted 2. And then in the Krebs cycle, we made 2 more, so we've made 4. So now we're going to move into, and there's a summary of that slide, the electron transport chain. And this is where the money is made. Now we're at the Federal Reserve, and we're just printing cash. And that's what happens here at the electron transport chain. So this happens on the intermitochondrial membrane. On the outside here, we have the intermembrane space, and on the inside, we have the matrix. The Krebs cycle occurs in the matrix. So we've got these NADH and FADH2 molecules just popping out right where we want them to be. Looks similar to the photosynthetic electron transport chain, and it's a similar design. Now, instead of using light, we're going to use a few different things to drive this reaction. There's a huge difference in electronegativity on these proteins, these transmembrane proteins. That's going to cause those electrons to be pulled across naturally. And when those, well, let's start from the beginning. So the NADH, how are we going to use that in this process? Well, you can split NADH and the electrons can be released. When the electrons are released, they go to the more and more electronegative proteins. So they'll start over here, and then they're naturally attracted to the next protein in the chain because they get more electronegative as they move across. Now, in these transmembrane proteins, there's a hole going all the way through that hydrogen can fit through, these protons. Well, when the hydrogens are coming off the NADH and the FADH2, so you've got tons of hydrogens floating around in here, just like we saw with the photosynthetic electron transport chain. Well, when the electron moves through the protein, it makes the protein negative, and it attracts these positive protons. So while the electron's in that pathway, the proton zooms across trying to catch the electron, But by the time it gets there, the electron's gone, but the proton gets trapped on the other side. And this keeps occurring with all these transmembrane proteins. Protons get zoomed through, and then they're stuck on the other side. So the intermembrane space swells up with protons as these NADH and NADH or FADH2 are being split. All of those protons are getting pushed to the other side. The electrons are zooming across. That's why we call it the electron transport chain. And the intermembrane space is swelling up like a balloon with all these protons. And just like the photosynthetic electron transport chain, all we need to do is make one opening so that the protons can only go through that one opening. And that opening happens to be ATP synthase, which takes ADP and phosphate, slams them together to make adenosine triphosphate. A ton of ATP. Well, where do those electrons go? We looked at that with the photosynthetic electron transport chain. At the end of this pathway, the electrons need somewhere to go as well. So remember we said oxygen was a necessary ingredient. You got to have it for aerobic cellular respiration. Well, the reason you need it is because at the end of the photosynthetic electron transport chain, you need an atom that is more electronegative than the most electronegative protein in the electron transport chain so that that electron will be pulled off the electron transport chain onto oxygen. And that's what happens here. It moves from more increasing electronegativity until, boom, it finds oxygen at the end, pulls that electron off, makes oxygen negative, which attracts it to some of the protons. And if oxygen, O, and two H's come together, you end up with water. So water is a product of cellular respiration due to the electrons attracting to oxygen, which is the final, it's known as the final electron acceptor. It pulls off those electrons, and now it becomes negative. It's attracted to H+, and it makes water. So we're getting a bunch of water, getting a bunch of ATP. So to summarize aerobic respiration, we're going to have 10 NADH and 2 FADH2 are going to be transported in to this pathway. And look what's made, 32 ATP. That's a lot of ATP. And of course, we're going to be releasing water as well. So here is where the money is made. Another name for this is oxidative phosphorylation. I think, why would it be called that? Well, oxygen, phosphorylation of ADP to make tons of ATP. So it kind of just summarizes the whole process there. So again, if we add everything together, 2 from glycolysis that we netted, spent 2, made 4, net 2, 2 from Krebs, 32 from ETC, we're going to make 36. Some cells are more efficient and can actually make 38. So what happens if our cells do not have oxygen? Well, they can do a process called anaerobic respiration. Some organisms always do this. It depends on what organism that we're talking about. Some organisms can do both. If we're talking about bacteria and fungi like yeast, or maybe we're talking about our bodies, if we're deprived of oxygen, our bodies will do lactic acid fermentation. But if we have oxygen, then we'll do regular aerobic cellular respiration. So it just depends. Some organisms will undergo anaerobic when oxygen levels are low. So that would be like us, for example. So the processes here are only 2. Glycolysis is going to happen. That's at the beginning of all respiration pathways. And then for anaerobic respiration, after glycolysis, it's going to move into fermentation. And it's either going to make alcohol or it's going to make lactic acid. So if we're talking about alcoholic fermentation, then we see that in yeast is the most common example. And that's what's used to make beer and wine. So it only involves glycolysis and then alcoholic fermentation. The number of ATP produced in glycolysis was 2 that we netted. And the number of ATP that will be produced in fermentation is also 2. So if you look at that, we're only going to get 4. So not super great, not very efficient. In humans, we cannot survive off of anaerobic respiration. Some of our cells can for a very short period of time, but overall, we're going to have to do aerobic respiration to survive. So here are the reactants and the products for anaerobic respiration. So for glycolysis, of course, we're going to put in glucose and spin to ATP. And we still make 2-pyruvate, 2 net ATP, 2 NADH. If we're doing alcoholic fermentation, we're going to plug in those 2 pyruvates that came from glycolysis, and they'll be used to make alcohol, or known as ethanol in this case, CO2, and 2 ATPs. So alcoholic beverages are made by doing this. It's a very simple process. We'll actually do this in the lab. We won't make wine or beer or anything, but we're just going to take yeast, mix it with some sugar water, and then cover it up so that it has to go through anaerobic respiration. And then we just see the bubbles that will be produced, because CO2 is a byproduct of fermentation. And we'll measure a column of bubbles. And we're also going to have a balloon attached to it, and we're going to see how that balloon swells up as those yeast produce CO2 through anaerobic respiration. So if grapes are used, if the sugar from grapes are used, they call that wine. If it's grains, that's how they make beer. And then honey, more in the past than it is today, was used to make what they called mead, which is an alcoholic beverage, because it came from fermentation, and alcohol is a product of fermentation. Lactic acid fermentation is another type. So depending on the organism, they might do lactic acid. This would be more for humans, or for yeast, they do alcohol fermentation. But we're still only going to make 4 ATP. So here we look at lactic acid fermentation. Same thing, reactants and products for glycolysis. And then those pyruvates plug into lactic acid fermentation, and it's going to make lactic acid and 2 ATP. This is what our bodies do when you work out really hard for a long time. You deprive your muscles of oxygen. Well, they're going to do respiration, but now they can't do aerobic, so they do anaerobic. And humans will do lactic acid fermentation, in which case they'll make lactic acid. That will build up in your muscle tissue, and that's what makes you feel sore the days after working out really hard. Here's yogurt, which is an example of lactic acid fermentation, and just the process by which they make it. Basically, they get it really, really hot, kill everything. Then they cool it down, and they mix in some bacteria that will do lactic acid fermentation. So then they get it at the right temperature. It will metabolize those the lactose in the milk, and will convert it into a lactic acid, which will make that nice sour flavor. That's an acid. It's a lactic acid that tastes nice and sour to create that desired flavor of yogurt. So it has a live culture of bacteria in there that is doing the work to make it taste that desired yogurt flavor. And that is all for this lesson.